Today we will be discussing the process of DNA replication. DNA replication doubles the chromosomes in the nucleus during S phase of the cell cycle before mitosis or meiosis, and takes slightly less than an hour to complete. DNA replication begins when the enzyme topoisomerase cuts the DNA, causing it to unwind from the double helix. The enzyme helicase then breaks apart the hydrogen bonds connecting the base pairs, separating the anti-parallel strands to create a replication fork. Single-stranded DNA-binding proteins then attach to the sugar phosphate backbone to ensure that the strands do not snap back into the helix. This results in the formation of two template strands. The top and bottom strands produce the leading and lagging strands, respectively. The leading strand runs from the 3' to 5' end, while the lagging strand runs from the 5' to 3' end. This is important because DNA can only be synthesized in the 5' to 3' direction. Beginning with the leading strand, the enzyme primase, a type of RNA polymerase, lays down the RNA primer. This molecule is about 12 nucleotides long, with a hydroxyl group on the 3' end. After the primer is attached, the enzyme DNA polymerase III attaches and lays down complementary base pairs to the 3' end of the primer. The DNA nucleotides link to the hydroxyl to form a phosphodiester bond. A protein called the sliding clamp holds polymerase III in place. Since the template runs from 3' to 5' and DNA is antiparallel, polymerase III synthesizes the new strand of DNA seamlessly in the 5' to 3' direction. Seamless synthesis cannot occur on the lagging strand because it runs in the 5' to 3' direction and the synthesized strand runs antiparallel. To begin, the template strand must be pulled in towards the replication fork so synthesis runs in the correct direction. DNA synthesis must take place in multiple steps. Primase lays down an RNA primer, and DNA polymerase III synthesizes the new strand. These steps are repeated, resulting in fragments of replicated DNA attached to RNA primers. These fragments are called Okazaki fragments. Once Okazaki fragments are made, DNA polymerase I replaces the RNA fragments with DNA nucleotides. The enzyme DNA ligase causes phosphodiester bonds to form between individual fragments, connecting them as one strand. However, in germ cells, synthesis of the lagging strand is not complete. The telomere, the end of the chromosome, has not been replicated since DNA cannot be synthesized without a primer. If this is not replicated, then the chromosomes will eventually become shorter and disappear. But the enzyme telomerase saves the day. It has its own set of RNA and uses this to extend the template strand so synthesis of the lagging strand can be completed. The completion of synthesis results in two identical DNA molecules. The synthesis of DNA is bidirectional, meaning that it occurs in both directions at the same time. However, this process does not occur without errors. Errors within DNA can result in life-threatening mutations, so it is important to catch them. There are two main components to error detection: DNA polymerase III proofreading and mismatch repair. The rigid structure of DNA helps prevent errors in synthesis. The complementary base pairs are energetically favorable and have distinctive shapes, making it apparent when an incorrect nucleotide is added. When an error is detected, polymerase must stop, remove, and replace the nucleotide before it can continue. Replacement of incorrect nucleotides after synthesis completion is called mismatch repair. A series of proteins removes a section of DNA, including the incorrect nucleotide. They then use an existing strand as a template to replace the section. These mechanisms result in an error rate of 1 in a billion nucleotides, a remarkable feat.